All right, so this is chapter 1, a brief history of microbiology. Microbiology is pretty cool because it has I wouldn't say, it's one of the newer sciences, right? We haven't had the technology to really do microbiology except for the last maybe like 250 years. So it's a very contemporary science, a very contemporary field of biology. And there's even new ones that are based off of microbiology that are going to be quite interesting as well. So what does life really look like, right? A lot of people were kind of curious about this, and one of the people that did this was Antony von Leeuwenhoek, right? So basically he started making very simple microscopes. What he used was essentially a glass bead in a kind of a holder where he'd have some specimen on the tip of this, pretty much a needle, right? And so he would have to make a new microscope for every specimen, right? Luckily nowadays, we get to change slides, right? It's a little bit easier, right? So he examined water and visualized tiny animals, fungi, algae, and single-celled protozoa, and he called those animalcules, like molecules, but with animals. And so by the end of the 19th century, these organisms were called microorganisms. That's what we do. So this is a painting of him doing this. This is the one you'll find in every book that talks about microscopy at all. They're like, this is the father of microscopy. He's also considered the father of microbiology. So a lot of people call him Micro Daddy. Thinking about putting that on a shirt and selling it. This is a example of what it is. So it's very small, and I've seen one or two of these in museums and stuff like that. But you can see how, you just have to get it in the right spot and all that kind of stuff. So that's what he was looking at. This is kind of something about what he would see, right? So we've got some algae here, right? We've got some protozoa. We've got some multicellular animals here, right? So there's a lot of different life that goes on in here, and he was one of the first to discover it. So one of the things when you discover something in biology is you have to classify it, right? We have this tree of life, and we like to make sure that it fits on that tree of life. And so this started with Carolus Linnaeus, right? And he developed this taxonomic system that we use today. And Leeuwenhoek's microorganisms basically fall into these categories. So bacteria and archaea, these are actually domains, right? Right. So these two are domains. Fungi, protozoa, algae, and small multicellular animals all are going to fall into the eukaryotes, right? So there's a lot of diversity here that we're going to go over in microbiology just based on the sheer number of different microorganisms that we see. So bacteria and archaea, they are unicellular. They lack a nuclei, right? That's what it means to be a prokaryote, right? That's kind of the definition, right? Archaea, so really where we find archaea are in very hostile conditions. And a lot of scientists believe, and there's a lot of evidence that supports that belief, that these were kind of primitive bacteria, right? And bacteria kind of figured a little bit more out, and then the eukaryotes came along and figured a little bit more out, and that's how life kind of came about. Bacteria and archaea are both going to be much smaller than eukaryotes. So I've got a picture in here that I'll show you here in a little bit. They're about 100 times smaller, right? So if you look in a microscope and you're like, oh, I have trouble seeing things in the microscope as it is, it's going to be a little bit harder for you to identify and find the bacteria. Luckily, we have better microscopes in micro, so it should be a little bit easier for that. We find them everywhere, right? There are bacteria living on your skin. There are bacteria living on the desk in front of you, on the pen you use, on the things you drink, on the things you eat, everywhere. Okay? If you ask me, yeah, germs are everywhere. Get over it. Lots of people are like, wash my hands, hand sanitizer. I'm like, we're all gonna get it someday. Anyway, they reproduce asexually, so they divide, right? They don't have any type of vertical gene transfer from like parent to child. However, their gene transfer does allow for horizontal gene transfer, which is from bacteria to bacteria. And so that's kind of a really interesting topic that we'll talk about later on in the semester. Bacterial cell walls contain peptidoglycan, although some of them will lack cell walls. Not very many of them do. The archaeal cell walls are composed of polymers other than peptidoglycan, but I don't think that they use chitin, because that is almost exclusively for fungi and for, what's it called? Invertebrates. Sorry, I'm blanking. Like bugs and stuff like that. Bugs and shrimp and all that. Oh yeah, so here's what I was talking about. So this is a picture of, or a micrograph, a light micrograph, of 2 human cheek cells. Right, there's one. And there's the other one. Right? The bacteria on this image are right here. And each one of these is an individual cell. That's how much smaller we're talking about. Okay? So they're even smaller than the nuclei of the eukaryotic cell. So, like I said, they're very small, hard to see. It's one of the reasons why we didn't really understand a lot of this until later. All right, so continuing on into fungi. So they are eukaryotic. They will have membrane-bound nucleus, right? They obtain their food from other organisms. So they are saprotrophs, I believe is the term for it. And they possess cell walls. Fungi are in this weird kind of not completely plant, not completely animal kind of thing. People who study fungi, they're called mycologists, right? They're really fun guys. Anyway, so molds, these are multicellular. They grow as long filaments, right? And they reproduce by sexual and asexual spores. So this is an example of a mold. And this is an example of a yeast. It is a single cellular organism. However, you will, a lot of times will find them in some type of colony. Protozoa, these are really cool. These are fun, these are just fun to look at. So these are single-cell eukaryotes, similar to animals in nutrient needs and cellular structure. Generally, they live freely in water. Some are going to live in animal hosts. There's very good chance that you have a couple of these floating around in your gut right now. That's just the way it is, right? Definitely a lot of places in your body you don't want these, like especially your brain. But there's also a lot of places where they're completely harmless to you. They do reproduce mostly asexually. There is some sexual reproduction, so you do get some genetic diversity eventually. Generally, the life cycles of these are, they have like multiple different life cycle kind of pathways that they can take, and you'll have some that have a sexual phase and an asexual phase, right? And so depending on nutrient availability and conditions, they will either do one or the other. Right? Most are capable of locomotion, so they can move around. They can do it through pseudopods, which are these little feet-like things. They kind of just stick them out, and they kind of come closer to it, right? Or more like this, like. Right? I'm not good at the robot, but it's like that. So those are really fascinating to look at when they're alive under a microscope. So hopefully we'll find some of those when we do like water samples and stuff like that. Cilia. So you guys have heard of cilia before if you've had AMP1. And so this is going to be another appendage on the outside, and they go like this. You know, they're just little hairs kind of that push them along. And then flagella, these are extensions of a cell that are fewer, longer, and more whip-like than the cilia. So you can see the flagella here, and then you can actually see the cilia on here pretty well. At least I can on mine, right? And so then you see the flagellum as well. So next we move on to algae. These can either be unicellular or multicellular. They are photosynthetic. They can make their own food, right? They have very simple reproductive structures, and generally they go asexually. But there is some ways to get genetic diversity into that. They are generally categorized on the basis of pigmentation and composition of cell wall. So these are just, like, I love looking at these because they're gorgeous, right? These are some of the cooler slides you get to look at, like in biology and stuff like that. They're a lot of fun to look at. So moving on from that, we have other organisms of importance to microbiologists, parasites. We'll spend a little bit of time talking about parasites. Yeah, there's a lot of parasites out there. And after you learn about parasites, you really watch what you eat. Anybody like snails? Escargot? Good. Because I wouldn't eat it. And then viruses, right? Viruses are quite interesting. There's a very good debate that we could theoretically have whether viruses are considered living or not, right? And the consensus that a lot of people have come to is they're not living, but we couldn't live without them at this point because there's been a lot of gene transfer from either different species or different things. In fact, I want to say some ancestor of the common deer now, what is it, white-tailed deer or whatever. That whole family ended up basically became mammals because of a virus, right? They used to not be that way. So, fun facts with Professor King. Now, the first time to tell me why, right? So some people consider Leeuwenhoek the father of microbiology. Why? He invented microscopes. He's one of the first ones to look at it, right? Pretty good one. Yeah, I'll take that. Anybody else? Okay. Yeah, that's why. You could probably draw another one in here and be like therapists. Right. So anyway, Backstreet Boys memes, because those are throughout the entire book. All right, so the golden age of microbiology. I probably should have looked up what years this was. I can't remember. But really late 1800s through the mid to late 1900s. God, I feel so old saying that now. Mid to late 1900s. I was born in the 1900s. Some of y'all weren't. Anyway. So scientists search for answers to the question, is the spontaneous generation of microbial life possible? What causes fermentation? Right, for lots of various reasons, mainly for booze. And what causes disease, right? I think this is probably one of the more important things that microbiology has done for us. And then how can we prevent both infection and disease? So, does microbial life spontaneously generate? No. But how do you prove that? Right? What's the idea, right? So philosophers and scientists argued about this, right? So in the past, they thought everything came from asexual reproduction or sexual reproductions, or even from non-living matter, right? Okay. Generally, you have to have a spark. There's some other experiments that kind of show if you put the right concoction together in a lightning storm, it'll turn in and start making proteins and stuff like that, which is pretty cool. So this is Aristotle, if you don't know. I'm sure you guys have met him before. He's dead. Most of the people we're going to talk about today are dead, by the way. So he proposed spontaneous generation, so living things can arise from non-living matter. That was his idea. He was like, well, we came from somewhere, so where did we come from? Well, we came from nothing. We came from matter, right? So the first guy who kind of looked at this was Reddy, right? And so he was like, okay, so decaying meat, you guys ever thrown meat away and then like let it sit in the trash can for a little while? What does it attract? Flies. Flies make maggots, right? So when decaying meat was kept isolated from flies, maggots never developed. Right? So they took an open flask, they put a piece of meat in it, right? Hopefully they didn't use a nice steak like it's showing. Hopefully they used some garbage cut of meat, but you know, whatever. So when they put a cork on the top of it, nothing developed, right? But when they covered it with gauze, they got maggots on the top of it, but not necessarily on the meat, right? Because they couldn't get through there. They couldn't lay their eggs into the meat, right? So as a result, scientists began to doubt Aristotle's view. So one of the things I want you to think about, and we'll talk about it a little bit more, is, okay, Aristotle had a hypothesis, right? I don't know if he did any tests of that hypothesis, right? If he did, I'd be interested to see. But these people were like, hey, I don't think your hypothesis is right. Let me design an experiment to see if your hypothesis holds up, right? Because if Aristotle's hypothesis held up, what would happen? The maggots would have been in the sealed one, right? So they just arise from nothing, right? So he designed this experiment to kind of see this, right? And so we're kind of just like, okay, well, where are these things coming from, right? We're not really sure yet, all right? So the next guy to do this was Needham, right? He was like, okay, what's going on, right? So scientists agrees that large animals could not arise spontaneously, but believes that microbes could, okay? So we're kind of getting to where we're like, okay, yeah, large animals, humans, you know, anything that's bigger than what we can see it with the naked eye, sure, they arise from other things, right, or other beings of that species, right? Needham's experiments with beef gravy and infusions of plant material reinforced this idea. So what he did was he had an open flask with this beef gravy and plant material or whatever, and he heated it up. And then he let it sit there. And then it turned cloudy. And then he's like, hey, look, spontaneous generation, right? He didn't put a cap on it, right? So we kind of are taking these little baby steps as we're doing this. Now, obviously, we're in the 1800s. Like, if you're thinking about it now and you've lived life, you're like, oh yeah, when I throw stuff in the trash or when I leave something uncovered, on the counter, yeah, things start growing in it, right? Even if you put it in the fridge, a lot of times things will start growing in something, right? So, continuing on, Spalzani, his experiments contradicted Needham's findings. You're like, wait a minute. Yeah, they don't just spontaneously generate. They got to come from somewhere, right? So Needham failed to heat the vials sufficiently to kill all microbes or had not sealed them tightly enough, right? So he was like, you may be right, but you may be wrong. Is that a song? You may be wrong, but you may be right. Yeah, I think so. I'm sure Siri could tell me. Right. So microorganisms also exist in the air and can contaminate experiments. Believe me, they do. When we do ubiquity here in a week or two, I'll just leave it played out all class, and then I'll seal it up, and you'll see stuff start growing on it. All right? That happens. So spontaneous generation of microorganisms does not occur. All living things arise from other living things. And so critics said the sealed vials did not allow enough air for organisms to survive, and that prolonged heating destroyed the life force, right? So Spalzani went around and he said, okay, we're going to boil this gravy, and then we are either going to cap it, right, or we're going to leave it open, right? So he's like, okay, let's boil it. Let's make sure everything's dead, right? So that's what he did. Then he did these two, right? So the gravy over here that's been capped is free of microorganisms. This gravy is teeming with microorganisms. Okay? So there's a lot of them in there. So this leads us to Louis Pasteur, right? So he performed experiments with the swan-necked flask, which is this really interesting flask here that's quite expensive if you're ever curious about how much they cost, because this little tube is hard to put on things, and then shipping it is quite difficult. All right? So when the flask remained upright, no microbial growth appeared. Right? So they heated it, right? And then they left it upright. Nothing happened. Right? But when the flask was tilted, dust from the bend in the neck seeped back into the flask and made the infusion cloudy. Right? So right here, air can move in and out of the flask, right? But because of the extra turns and stuff like that, the bacteria and the dust and all that other stuff wasn't going to get in there, right? So the dust would settle right here, right? And if you tilted that flask, right, some of that broth in there, whatever was growing in there, or whatever they had in there, would get some of that dust in it, and then you'd introduce these microbes. So really this all kind of helps us to describe the scientific method, right? So the debate over spontaneous generation led in part to the development of the scientific method, right? People were doing this and have been doing this for millennia. They just didn't know what it was and didn't write it down that they called it this, right? Somebody at one point figured out if you rub two sticks together long enough, it creates fire. We have had fire for how long? Like I never took an anthropology class, but we've had fire for quite a while, right? So we've been doing this for a long, long time. We just haven't really named it and put anything on it, right? So an observation leads to a question, right? That's the big thing, is questions. One of the biggest things, if you're going to be a scientist, is having this inquisitive mind, right? And unfortunately, in a lot of education, this inquisitive mind is kind of beaten out of the kids, for lack of a better term. You go find a kindergartner, they will ask you, why? Why is that happening? What's that? How's that? They'll ask you 100 questions in an hour, right? But as you get older, you stop asking those questions. You stop asking why. You stop being curious about your world. And we kind of lose that curiosity. And when you lose that curiosity, that's when you stop really learning about science and the world around you. But when you have these questions and you're like, okay, this is what I think the answer is. That's your hypothesis, right? And so we've generated our hypothesis, and then we test our hypothesis, right? So somebody said, hey, if I rub two sticks together long enough and get them hot enough, it will start fire. And so they generate. Will doing this lead to a fire? So they rubbed two sticks together. They got fire, right? The results either prove or disprove the hypothesis, right? And so accepted hypotheses generally lead to a theory or law, right? And when we talk about theories are backed up in science with tons of data, right? With lots and lots and lots of experiments that said, hey, yeah, this works for me too, okay? Laws, like the law of thermodynamics, Newton's laws, stuff like that. Most laws in science, they are going to be very pretty much unchanging. Like, we figured it out. Like, we know that, if you are under the force of gravity and you drop something, it's going to fall. It's going to fall towards the bigger object, right? But, the theory of germ theory, right? Do microbes cause disease, right? And then Koch, I think it's Koch for this one, has its postulates, right? But we can't satisfy every one of the postulates because it's unethical for us to infect people with stuff to see if they get sick. Yeah. Not that it hasn't been done before, but like I said, it's unethical, right? So disproved hypotheses are rejected or modified, right? So the idea of science is you're searching for the truth, right? And so when you reject the hypothesis, you're like, okay, that's not the explanation for this, right? You modify it. You say, okay, well, what is the explanation for this? If I water my plants, they will grow. Well, if your plants don't grow when you water them, why? Well, it's a succulent. It only needs watering very little, but you watered it very much, right? So you killed it. Okay. So this is kind of how it goes. There are so many different representations of this. I've seen it represented as a circle. I've seen it represented as a linear process. I've seen it represented in a flowchart like this. Pretty much the scientific method boils down to you have a question, you form that hypothesis, you test that hypothesis. If it's right, cool, you confirmed your hypothesis. You should probably do some more testing to make sure it's actually true, not just a fluke, right? But if it's not, you either reject it or you modify it and you start over, right? That is the idea, right? Very, very few things that we will ever do, probably in our lifetimes, will ever become a theory or law, right? We've discovered a lot. So we're getting to the point where we're kind of running out of that. So, what causes fermentation, right? That's our question. Spoiled wine threatened the livelihood of many grape growers, right? Anybody ever had corked wine? Or not corked wine. Is it corked? Is that the name for it? When your wine tastes terrible because it's like gone bad. Yeah, I don't drink wine, so that's one of the ways that it can happen, right? The cork can actually infect it with something, right? So wine producers funded research of methods to promote the production of alcohol and prevent spoilage during fermentation, right? If you let this ferment too long, it'll probably turn into what, vinegar, I think, and that's why it's that. Anyway, the debate over the cause of fermentation reactions was also linked to the debate over spontaneous generation. So Louis Pasteur, let's go back to him, right? You've probably heard this before when you've heard about pasteurization. Things are pasteurized to keep you safe and keep you healthy, right? You will hear some people who exist on the internet that say, oh, you should drink raw milk directly from the cow. That's the way to do it, right? And, you know, I've read anecdotes about people who lived on farms and they drank raw milk. And they said, well, man, someone in my family was just always sick. You know, if it wasn't dad, it was mom. If it wasn't my brother, it was me, you know. And then they grew up, went off to college, started drinking pasteurized milk, and they're like, I never got sick anymore. We could develop a hypothesis off of that question, right? Don't drink raw milk is the moral of the story, right? If you do so at your own risk and your own peril, and I will probably look at you funny, especially if you've taken this class and you say, I still want to drink raw milk. I think it's good for me. Yeah, there's also people that are into like raw water, and that's even worse. Like, just don't.
What does that even mean?
They go to like a stream or a pond and they get it. And I promise you, after we look at water samples in this class, you will 100% make sure the water you put in your body is clean. Yeah, I mean, yeah, for most lakes, yeah. You don't swim in the lakes around here. There's gators, that's why. So, all right. So Pasteur conducted a series of experiments that addressed the cause of fermentation. So here are his experiments. So spontaneous fermentation can occur, right? The flask is uncovered, right? So on the first day, they boiled it. They boiled the grape juice. They heated it sufficiently to kill all the microbes. Then they sealed it, right? Day 2, right? It's still sealed. No fermentation. Juice remains free of microbes. So you reject the hypothesis 1, right? Air ferments grape juice, right? That's the hypothesis 2, right? So flask remains open to air via this curved neck, so no fermentation occurred, right? The juice remained free of microbes, so we reject this hypothesis, right? So bacteria ferment grape juice into alcohol, so juice in the flask is inoculated with bacteria, then sealed, right? It's heated up. Bacteria reproduce, acids are produced, modify hypothesis 3 because the bacteria ferment the grape juice into acids. Acid isn't very tasty, right? And then yeast ferments grape juice into alcohol. So juice in the flask do the same thing, right? The yeast produce the alcohol, right? And we accept hypothesis 4 that yeast ferment grape juice into alcohol, right? Yeast is very useful, right? If you want to make your own alcohol out of your own juice, you can do that. There are kits out there on the internet that will allow you to do so. It's somewhat tasty. You can turn like Mountain Dew into wine if you really want to, but I just wouldn't suggest it. Like, it's probably not going to taste very good. Yeah. Anyway. So Pasteur's experiments led to the development of pasteurization. They named it after him. How nice, right? So this is a process of heating liquids just enough to kill most bacteria. And a lot of times what they'll do is they'll heat it up at a standard pressure, right? So they will seal it in some type of vessel, and they will heat that vessel up so that you raise the pressure, so the bacteria don't like it. You also raise the temperature. The bacteria really don't like the combination of both of those. So that way they can essentially boil the milk at a much lower temperature and not denature the proteins, right? So this began the field of industrial microbiology, which is the intentional use of microbes for manufacturing products. I think we'll get into it, right? So foods and beverages, right? Cheese. I don't know if anybody likes good cheese in here. I do. So flavoring and ripening produced by bacteria and fungi. Flavors dependent on the source of milk and the type of microorganism. Alcoholic beverages, if you're an adult and you partake, yeah, kind of tasty, especially if you mix them just right. You know, a dark rum, a light rum, some orgeat, some lime juice, and you know, a little mint sprig to garnish, and you know, you got yourself a Mai Tai. So yeah, probably shouldn't say that on the recording, but you know, whatever. It's a dry campus, by the way. Don't be bringing stuff on here. Yeah, soy sauce. Very tasty, right? Especially if you do this in the very traditional method. Very interesting to watch. Vinegar, yogurt, sour cream, artificial sweetener, right? So these amino acids are synthesized by bacteria from sugar. And then we essentially wash out the bacteria, purify it, put it in little packets, and then you eat it. And it tastes sweet. And then bread. I'm like Oprah. I love bread. Only a few of you get that. Other products, antibiotics, right? So we use bacteria to make antibiotics. Same thing with fungi. Human growth hormone as well as human insulin. So all human insulin for human use is now produced in bacteria instead of being produced in either cows or pigs, right? And when they first switched over to that, a lot of people were not very happy about it, right? They were like, oh no, it's not the same, blah, 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 blah. And like now they're like, oh yeah, it was fine. Right? I remember watching a news article. It happened in the early 2000s. And they were like freaking out. And they're like, I've got like a whole fridge full of it. And I'm like, this stuff expires, right? Like insulin goes bad. Like sure, you can use expired insulin, but probably shouldn't. Right? But, what do I know? Laundry enzymes, isolated from bacteria, like one that I think of the most, like the pet sprays. Like your pet peed on the carpet and you spray it on there and it smells kind of funky and then it doesn't smell funky anymore. Yeah, that's fine. Vitamins, a lot of times, are isolated from bacteria. Diatomaceous earth, right? Used in polishing compounds. It also will help you keep bugs away in your garden, right? It's a very good way to control bugs because it slices them up like crazy. So, fun facts from Professor King. Pest control chemicals, right? So, insect destroying bacteria. Bacillus Bacillus thuringiensis. They've infected corn with this bacteria, and when caterpillars and other things that eat the corn come in and eat this corn, right? It actually produces little crystals inside of their guts and eviscerates their guts, and the bugs die. It's a GMO crop, right? But we don't have to use a whole bunch of pesticides on them. So, and then drain opener, protein digesting and fat digesting enzymes. Yeah, that's, be careful with drain cleaners. Really not safe. All right, so what causes fermentation? Continuing on here. Buckner's experiments demonstrated that fermentation does not require living cells, right? So what did Buckner do? He basically isolated the enzymes from inside the cells, and he showed, hey, yeah, you can just use the enzymes. Right? You don't actually have to use the bacteria to do it. And so, Buckner's work actually began the field of biochemistry. Right? So this is kind of the start of that. So Pasteur developed the germ theory of disease, which we will spend quite a bit of time talking about in this class. And some diseases are caused by specific germs called pathogens, right? We can think of a few of them, right? I'm sure you can think of at least one virus that is caused by a pathogen, like the flu. So we're going to see a lot of pictures of people. Most of these people aren't happy, right? And I was talking to my wife about this yesterday, and I said, I don't know if it's just because it was like in the old days and they didn't smile in pictures, or if it was they were just so tired of everything that they didn't want to do anything anymore, right? So, grain of salt, just, if you think funding's hard to get to now for a science lab, then yeah, probably even harder then. Right, so this is Robert Koch, or Koch, I think it's Koch, anyway. Koch, I think it's Koch. Anyway, whatever. He studied the causative agents of disease or etiology, demonstrated that bacterium causes anthrax, right? And he examined colonies of microorganisms. So he was responsible for a lot of stuff. So simple staining techniques, the first photomicrograph of a bacteria, the first photograph of a bacteria in diseased tissue, right? Techniques for estimating bacterial number in a solution, which we'll get to do very similar to what he did. Use steam sterilization of growth media. We'll do that. You won't do that, but we'll prepare it, and then it will be steam sterilized. Use of petri dishes, laboratory technique to transfer bacteria, and bacteria as distinct species. So there are differences between this bacteria and that bacteria, right? So he did a lot. He really did that. This next one, Fanny Hess, her husband was a microbiologist, and she, I think, was just helping out in the lab. There wasn't a whole lot about her mentioned. But what she did was she said, hey, instead of using gelatin for your growth medium, you should use agar because it's more stable at higher temperatures, right? What happens if you get Jell-O hot? It melts, right? And so what happens if you get agar hot? Well, it melts, but it melts at a much higher temperature, right? And so we'll find that out in lab. Right, so here we can see bacterial colonies growing on a petri dish. All of these are different bacteria, right? Even the ones that look like they're the same color, right? These are bacterial colonies on here, right? They could be different bacteria, they could be the same bacteria, right? There's lots of different things that go on there. So Koch's postulates, these are kind of ways that we can kind of identify and say, hey, these have to be satisfied for us to say that this microorganism causes disease. Right, so the suspected causative agent must be found in every case of the disease and be absent from healthy hosts. Can we do this ethically? Yes. Okay, we can take blood samples, we can take tissue samples from infected people, and we can take a look at that. The agent must be isolated and grown outside the host. We can ethically do this. It is not always possible to do this. Some bacteria and things don't like to grow outside of their host, right? Very, very difficult. In fact, one of the things I worked on when I was working in a lab, we were working on citrus disease, right? And the bacteria that causes citrus greening, Hunglong Bing, what is it? I can't remember the name of it. But anyway, up until probably a year or two ago, they could not culture that bacteria outside of the host, right? Which is either in the gut of the psyllid that transmits it or in the tree, right? When the agent is introduced to a healthy susceptible host, the host must get the disease. Is this ethical? No. Right? There are studies that have done this, right? In fact, with the COVID vaccines, there were a handful of studies that actually did this. They took healthy patients and said, yeah, we're going to pay you a lot more than the other people got paid to do this. we're going to dose you with the vaccine, let you develop an immunity, and then we're going to dose you with the actual disease. And they did that, right? Very, very hard to get approval to do this. And luckily, none of them died, right? And I think only one of them got sick enough to even need like medical attention, essentially. Can't remember how big the sample size was, but anyway, not something that's terribly ethical. Like, we can't infect somebody with a deadly virus to say, hey, did this cause this? Like, hey, here's some Ebola. You wanna do that? Yeah. All right, same agent must be found in the diseased experimental host. Once again, a lot of times this requires killing the host to, dissect and look for it. Probably not that ethical, right? So if we can satisfy all of these, we can say, yeah, that's the cause of it, right? But like I said, ethics rules are there for a reason. We don't want to be terrible scientists. So yeah, let's. All right. So the golden age of microbiology, I guess, runs from 1883 to whatever the end of this thing is. There's a lot of different ones on here, and I encourage you to make sure you read through this. Because I think it's a really kind of interesting timeline. And most of it is, like I said, in the last 200 years, right? So it's really kind of a cool thing there. You guys can barely see that. I'm sorry about that. All right, so traveler's diarrhea, bladder infections, right? E. coli, right? So, yeah. Anybody ever had traveler's diarrhea? It's awful, right? What about a bladder infection? Yeah, that's more common, right? A lot of people have had those, right? Let's see. Meningococcal meningitis. I know when I was on my way to college, I was told, you need to go get that vaccine. Yep, because you'll know about 3 hours before you're in the hospital dying from it. Yeah. Salmonella, tetanus, right? Tobacco mosaic virus, right? So this is the disease of the tobacco plant, and this is some of the first stuff in virology that we really saw. My former principal investigator, he actually discovered some stuff about tobacco mosaic virus, and that kind of rocketed him up in the virology world, which is pretty cool. So, gas gangrene, right? Bubonic plague, that's nice and fun. Shigellosis from Shigella. Yellow fever and African sleeping sickness, right? So all of these people are coming in and discovering these things and helping us understand, oh yeah, hey, if you get bit by the tsetse fly, you're going to get African sleeping sickness. Cool. Right. One of the biggest ones here is Graham, right? I don't remember his first name, but Graham. All right, so this is the most widely used staining technique, and it's one of the first steps to identify a bacterium, right? And so what it is you get purple, gram positive, and you get pink for gram negative, right? And so we'll get to do this in lab, so we'll get to stain some stuff, and you'll probably stain it horribly the first time, and by the third or fourth time you do it, you'll probably figure it out, right? It's really kind of a cool process, and to do it well takes a little bit of practice. So how can we prevent disease? Right? We understand now, and we're starting to develop this and say, hey, we can prevent disease, right? How do we do this? Well, Semmelweis, right? This guy right here. He said, hey, I think we should wash our hands in the obstetrics unit of this particular hospital. And the doctors laughed at him. Okay? He went to the midwives and said, hey, you guys want to wash your hands between patients? And they're like, hey, it doesn't sound like a horrible idea, right? So they did it. And so basically it was the midwives versus the doctors. And the midwives got the incidence of puerperal fever, essentially a fever you'd get from, it's some bacterial infection. Anyway. You get a fever and you die, right? And a lot of times the child dies too, right? So they were able to make it with the midwives washing their hands go from about a 20% incidence per chance per patient to a 2% chance per patient, just by washing their hands, right? The doctors were all pissed off about it and were saying, well, blah, 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 right? And they were so mad about it that they really hammered on Semmelweis, right? And was like, yeah, you're an idiot. We don't like you. You can't explain why it works, so we don't like you, right? Eventually, Semmelweis had a mental breakdown. He gets committed to an insane asylum by his colleagues, the ones that were like, you're crazy, you can't explain it, go screw yourself, right? And then about 2 weeks after he gets in there, he ended up getting beaten up by the guards and then ended up dying from a wound that became gangrenous. So, sometimes science is not as accepting as it should be. He could show, hey, this works, but I don't know why, right? The other doctors should have been like, I wonder why that doesn't, or why that works, and helped him out instead of saying, you're crazy, go die in an asylum, right? Yeah, anyway. All right, Lister, his antiseptic technique, so he was a surgeon. And if you read about him, he was not an exceptionally good surgeon. He was actually quite terrible at it. So the fact that he didn't even mention in a lot of these things is because of, well, he figured out how to do it without getting people sick, right? And so he was the kind of the pioneer here to a lot of the aseptic techniques that's used today, right? So a sterile field, making sure that your instruments are sterilized, using phenol to cleanse wounds, as well as preoperative sites and stuff like that, and all that kind of stuff. Nightingale, I'm sure you have probably heard of this woman. When I say that, Van Leeuwenhoek is micro daddy, this is nursing mommy. Just saying. You'll hear plenty about Nightingale in nursing school. And so she was actually a statistician, right? By training, she was a statistician. So she came in there and she started kind of doing a lot of things that were like cleaning up and making sure things were taken care of, and bandages were changed, all that kind of stuff, to kind of improve the outcomes, right? And so she kind of started this idea that maybe we should, take care of our patients instead of just sitting there and letting them rot. Whatever. So, John Snow. Oh, not this guy. Sorry. John Snow, he started to work on infection control and epidemiology. So, this guy, John Snow, right, there was an outbreak of cholera, I believe. Yeah, I believe it was cholera in his town, right? And he was like, I wonder what happened. So he ran around and he mapped it. Right? And so each of these little things, you can't really see it very well, but they're like little stacks, right? So each of those lines was somebody who got it, right? And, I can't find it. Where was it? I should have found it. Oh, here we are. Pump. There's another pump too. But anyway, what he found was this particular well was contaminated because at the time, people just threw their poop in the streets, right? And so people were getting this in these areas because they were being, their water source was being infected by essentially the sewage, right? And so that's kind of the beginning of epidemiology there. Edward Jenner, not to be confused with Bruce Jenner, he did vaccines, right? He kind of started this idea of vaccines in the field of immunology. Now, he wasn't the first one to do some type of vaccination. And when I say that, I'm saying that kind of cautiously because there's some other stuff that we'll talk about here in a little bit called variolation, and there's some other ways to inoculate people against something that isn't necessarily a vaccine, right? So what he did was he was like, hey, smallpox and cowpox are pretty similar, right? And so he went to the udders of cows and scraped some of those poxes off and the little sores, and then he would use a needle to kind of stick it into somebody, right? This is a statue of him doing it to his son, right? And, you know, I think that one of the things that, like, we've eradicated smallpox, right? We don't see smallpox anymore. And rightfully so. It was an awful, terrible, disfiguring disease, right? We've almost eradicated polio. Almost. There's a couple of pockets of it in South Asia, and I think a little bit in Africa. But we've completely eradicated smallpox, and I think a lot of people have forgotten that people got terribly sick and disfigured because of this. And it was a constant reminder of, hey, yeah, you could get smallpox too, right? There's a lot of vaccine hesitancy out there, but it's because a lot of times people don't get sick. We're cleaner than we used to be. We're living longer than we used to be, and one of the reasons we're doing that is we don't have as much inflammation from all of these environmental insults that we had, right? So, you know, I think if we saw diseases like this, or saw people and said, oh, hey, look, that person's suffering from the side effects of polio. You know, they can't walk anymore. They live in an iron lung, you know, those kind of things. I think we would have a much different kind of view on a lot of things because we don't see it anymore. Ehrlich, I think it's Paul Ehrlich, can't remember. His magic bullets, right? So he started the field of chemotherapy. And when a lot of people hear chemotherapy, they think of cancer, right? And while that's true, chemotherapy is used for cancer. Chemotherapy actually was first used and is still used for things like antibiotics. And so this was his grad student that actually discovered a lot of the stuff, but, you know, Ehrlich took credit for it. And so one of the things that they discovered was a treatment for syphilis, right? And this treatment for syphilis allowed them to treat someone, and then they were no longer infected with it. Great, right? Which means they couldn't spread it, right? And it worked. It worked for a while. It doesn't work anymore. If you get syphilis now, good luck. Yeah. We don't have a chapter on it, but we probably should have a chapter on bag it before you shag it. But, you know. Anyway, Lady Mary Wortley Montagu, right? She's got some interesting pictures here. She has a real interesting story. So she was a English aristocrat that got married off to some Turkish royalty or something like that. Kind of cool, right? But she actually did the variolation, right? So she looked at what people were doing in Turkey, and when people had smallpox in Turkey, that a lot of times they would come by people's houses, and then they would take a needle and they would take some of the smallpox, and they would put it on either their arm or their leg and do the poke you 100 times kind of thing, right? And this is what we call variolation. And so she's like, hey, this works. When you take a very small amount of the smallpox, hey, cool. Yeah, it's you're going to develop one sore, and then your body's going to take care of it. But when you get infected with smallpox, it's all over, right? So this is another way that they were able to do that, and she actually did it to her son. Their picture there in the middle picture. And she's like, look, it's safe, everybody. I'm doing it to my own son. So. Exactly. Yeah. I wouldn't suggest doing it now. You have a vaccine for that. I wish I had a vaccine for that because I got chickenpox twice. Usually get it once, right? So I'm going to have fun shingles is what I'm going to have. Right? The funny story about this was this picture right here. And I want to say this guy's name's Alexander Pope, but don't hold me to that, right? He professed his love for Miss Lady Mary Wortley Montagu, right? And this is her laughing at him. And this is not a picture of her. This is a painting of her. So she had to be so just laughing about this for so long that she's like, I need a picture of this hanging up in my residence. And so she commissioned this painting of her laughing at this guy who was like, I love you. And she's like, ha ha ha, I don't. right? Slay queen or whatever you kids say nowadays, right? So yeah, there's a lot of things that go on, right? What's interesting is, we see a lot of this stuff, and we're like, wow, this is kind of the beginning of a lot of science, right? We've looked at a lot of different fields of science that have basically grown out of microbiology. All right, so let's go through some of these. So these guys: infection control, epidemiology, taxonomy, basically microbiology up here, right? Pasteur for pasteurization and industrial microbiology, Buckner for microbial genetics, genetic engineering, etiologies from Koch's postulates, right? Virology from Ivanovsky, right? All of these other people really came through and said, hey, we can take this information and do something else with it, right? Which is really just cool, right? And so we're still seeing a lot of stuff that is, still very, very useful, like serology, immunology, and stuff like that. Like, we're still working on a ton of that stuff. None of this stuff is done, right? Which is what's really interesting about microbiology. Tell me why. Yeah. Anyway, so why might Nightingale be considered the mother of medical microbiology? Washing hands, cleaning, changing bandages. If something smells, you should probably clean it or change it. Yeah, kind of common sense things, right? So some other fields: so bacteriology, phycology, bacteriology, bacteria and archaea, phycology for algae, mycology for fungi, because mycologists are really fun guys. Haha. Anyway, protozoology, protozoa, parasitology, parasites, virology for viruses, right? Process-centered, right? So microbial metabolism, right? So this kind of centered around biochemistry. That's what essentially came out of that, right? Microbial genetics, the functions of both DNA and RNA. It's a lot easier to study bacteria because they grow much faster and they don't need as much growth medium and essentially care as a eukaryotic cell might need. Environmental microbiology, so the relationships between microbes among other microbes and other organisms and their environment, right? We are now learning about all of the things that are inside of our gut. And all of those things inside of our gut, yeah, they do a lot of things that we didn't know they did. Like the microbiome inside of you and on your skin and everywhere in your body is quite the Wild West of science right now, right? They've linked it to a lot of things, whether it's a causal link or a, what's the other one? Direct link. I don't remember off the top of my head. It's yet to be discovered for a lot of that stuff. Oh, correlated link. That's it. Right, so in applied microbiology, we have serology, right? So antibodies and blood serum, right? And also, so this is useful for a couple of things, right? When we had COVID going on and people were like, oh, I want the Regeneron, that was a serological kind of thing, right? They took antibodies out of one person, put them in somebody else. Or not out of a person, they had bacteria make them, right? But also as an indicator of infection, right? This is where they kind of got ELISA from. You guys have probably taken a COVID test, right? You swab your nose, right? You put the thing in the juice, right? And then you mix it around, right? Get it all off of there, right? And then you take the thing out and you stick it in the whatever, or you drop the drops on there, right? And then you wait and see, right? That's an ELISA test. That's what you just did, right? Immunology, the body's defenses against specific diseases. Epidemiology, the study of frequency, distribution, and spread. Etiology, the cause, right? Infection control, that's huge, especially in hospitals, right? You don't want more people getting sick or getting sicker while they're in the hospital, right? Chemotherapy. Bioremediation, this is actually pretty cool. I was reading something the other day where they had found a fungi that would basically take radioactive material and turn it into not necessarily less radioactive material, but safer radioactive material. So there's a lot of bacteria that are used for pollution remediation, especially with think they found some for like crude oil and stuff like that. So there's a lot of different ways that can be used. Public health, sewage treatment, as well as monitoring, water purification, control of insects that spread disease. You should be very glad they spray for mosquitoes around here, right? There's a couple of weird, scary mosquito viruses that are on the horizon. You don't want any of them. And then agricultural microbiology, using microbes to control pests, right? Food and beverage, yeah, heck yeah. You like your food to last a while when you buy it, right? There you go, right? A lot of people like, oh, I don't like preservatives in my food. Yeah, okay, neither do I. I like fresh food. I agree. Fresh food goes bad faster, right? Right, and so people are like, well, why doesn't it last like, a month like it used to? Well, we took out all the preservatives. So you got to eat it faster now, right? Pharmaceutical, the manufacturers of both vaccines and antibiotics, and recombinant DNA technology. So that's where we go in and alter the genetics of microbes to synthesize useful products, right? So there's a ton of microbiology that you can explore, and so I think it's definitely one of those things that's worth doing, right? So this guy, this is Cliver. I couldn't find a picture of Van Niel. So basically, biochemistry kind of arose out of all of this, right? So he began with Pasteur's work on fermentations and Buckner's discovery of enzymes in yeast extract. So he used microbes as model systems for biochemical reactions, and so the practical applications of a lot of stuff that he designed was herbicides and pesticides, diagnosis of an illness, of monitoring a patient's response to treatment, treatment of metabolic diseases, as well as drug design. Drug design is a big industry. In fact, what's scary about it is we've produced a ton of antibiotics, but we're really slowing down. There may have been, like in the last 5 years, maybe one new antibiotic, and in the 90s we were putting out like 10 every year, right? So that's really slowing down, and more synthetic ones are coming that way now, even though they could probably find a lot in nature too. All right, so how do genes work? Microbial genetics, microbiology, molecular biology, recombinant DNA technology, and gene therapy. So we'll talk about all of these. I'm running low on time today, so I may have to push this, but we'll see. All right, I didn't put pictures of all these people because there's like 100 of them, right? Avery, McLeod, and McCarty determined genes are contained in the molecules of DNA. Beadle and Tatum established that a gene's activity is going to be related to the protein function, right? So you guys probably remember, or at least should remember, DNA is what into RNA? Close. Right.
Wait, what did you ask?
DNA is what into RNA?
Like, do you mean the letters?
No, like transcribed.
Oh,
Yeah. And then to go to proteins, what do you do? What do you do? Translate. Because DNA and RNA are the same language, right? Just a slightly different dialect. And then you translate into proteins the language of amino acids, right? So the translation of genetic information into protein, right? The rates and mechanisms of genetic mutation, right? And to identify methods cells use to control genetic, right? So molecular biology really was to explain how things work inside the cell, which I think is really fascinating. I think it's really, like, if I could go back to school for anything, it'd probably be molecular biology, just to kind of figure out more of how the cells actually work. I think it's just fascinating. So Pauling proposed that a gene sequence could provide an understanding of evolutionary relationships and processes, right? And so this is one of the ways that we've started to change the taxonomy that we see. So as we sequence more and more genomes, we're finding out that different things are related, right? Used to we would look at their outward traits, right? Now we look at their genes, right? So this is one of the reasons why they have said, what's it called? Birds and reptiles are more closely related than we think, right? They're very, very closely related in that dinosaurs probably had feathers, right? So that's where they're getting that from. And to also identify the existence of microbes that have never been cultured. There have plenty of microbes out there that have never been cultured, right? Just wait until the permafrost in the far north and south melt, and we'll find out about a lot of microbes that we have no memory of, and our immune systems are probably going to be like, huh? And yeah, then we're going to die. It's all right. Weiss and Fox determined that cells can be categorized as bacteria, archaea, or eukaryotes, and cat scratch disease caused by an unculturable organism, right? So all of these different things have been discovered through biology. Recombinant DNA technology, so genes in microbes, plants, animals are manipulated for practical applications. If you've been to a pet store and seen the glowfish, that is a recombinant DNA technology in use in there. They use, I think for a lot of them, jellyfish genes for at least the yellow, and I think the green ones. The pink is coral. That color comes from coral. I want to say the yellow comes from coral as well, and then I think the blue comes from something weird, but the blue doesn't really work too well. The production of human blood clotting factor by E. coli to aid hemophiliacs, right? So we've done a lot of that, kind of using recombinant DNA technology for that. Gene therapy, so inserting a missing gene or repairing a defective one in humans by inserting a desired gene into the host cells. We've done this. In fact, they did a trial with sickle cell anemia in patients who suffered from that pretty severely. And they swapped out some of their bone marrow for a recombinant gene therapy, and they fixed it completely, right? 100% effectiveness, which in medicine is absolutely out of this world, right? You're lucky if you maybe get like 20 to 30%. Right, so what role do microorganisms play in the environment? Bioremediation, right? Living bacteria, fungi, and algae to detoxify things. Recycling of chemicals such as carbon, nitrogen, and sulfur. If you've ever had a fish tank, the nitrogen cycle is something you're very interested in, right? And a lot of times people will get a fish tank, they'll set it up, they'll put fish in it, and then the fish will die, right? And the reason why the fish died is because you have to have set up the nitrogen cycle before you put the fish in there. Because if you don't, the nitrites will kill the fish. But if you have the bacteria that turn nitrites into the harmless, or at least in freshwater, nitrates, you're golden, right? You're fine. So that. Sulfur, yeah, okay, there's some fun stuff with that. The causation of disease. So, obviously we're surrounded by microbes, but that's one of the things that we take a look at in the environment. So how do we defend against disease? Well, serology, that's the study of blood serum, right? Von Behring and Kitasato, existence in the blood of chemicals and cells that fight infection, right? So we'll learn about our immune cells. We'll look at some of them here in lab today. The immunology, so this is the study of the body's defenses against specific pathogens. And chemotherapy, Fleming discovered penicillin, which pretty much was very instrumental in helping us win World War II. USA, back-to-back World War champs, right? No. Yeah. Domack discovered the sulfa drugs. So if you ever hear, one thing, we shouldn't advertise prescription medication on the TV, but if you do watch TV, watch it, and you'll see, oh, blah, 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 you shouldn't take this if you're on an MAOI inhibitor, blah, 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 or sulfonamides. That kind of drug. Very, very similar. All right, so like I said, so this is the penicillium growing here. This is that zone of inhibition. We're going to do something like this in lab, if I'm not mistaken. And then this is the bacteria not growing around it, right? It doesn't want to grow close to it because it's like, wait, you make things that are bad, right? And so they were just like dosing our soldiers with penicillin because they're like, don't get sick. Here, go to a war zone, but don't get sick, So what does the future hold? Microbiology is built on asking and answering questions, just like most sciences, right? Or all sciences. The more questions we answer, the more questions we have, right? So science isn't done, right? It's never finished. It never will be finished, right? It's also kind of interesting. Every time we discover something else, we realize we know less and less about the world around us, right? So as you continue through school, if you go get a graduate degree or you go get a PhD or something like that, you'll find out how little you actually know about the world, right? And there's a lot of people who are experts in things, but they are expert in a very, very small subset of things, right? Which is, it's cool, it's useful. You know, we need those people in the world, but it's also good to know that you can never know everything, right? So tell me why, one last time, right? There are so many modern questions in microbiology related to genetics. Anybody got an idea why? Anybody? Yeah, I mean, it's where everything comes from, right? And so it's the basis of everything that we will study, right? So what makes E. coli different from, you know, Bacillus thuringiensis, right? It's the genetics, right? They're all producing similar proteins, but how are they using them, right? So very good. Don't forget to like, follow, and subscribe, and ring that bell icon to be notified whenever I post a new video.